Aditya, you may start whenever you are ready.
Yes, ma'am. Can I start now?
Yes.
Okay. Good evening, everyone. I will be presenting my project today. Uh, in my project, I will firstly explain the idea using a few slides, and after that, I will show the actual codes and the output so you all can see how it was implemented and done. Is my screen visible now?
Yes, it's visible.
Okay. So here's my project title. Before that, first of all, myself Aditya Tiwari. I'm a first year BTech Bioinformatics student at Sam University, Prayagraj. Today's my project title is Functional Sequence Characterization using Biopython. In simple words, we can talk about this project is about taking a protein sequence whose function is not known yet, and still we are trying to predict what it might do using bioinformatics tools named BioPython. This kind of analysis is pretty common in bioinformatic areas, research areas specifically, if I talk about, because many sequences are discovered, but yet their functions are not experimentally studies. So, the objective, or if I say the main objective of this project, of my project, was to analyze a hypothetical protein sequence and predict its possible biological function using computational methods. A hypothetical protein, what it is, it basically means a protein sequence which is available, but we don't know what it does or it might be what it might be. So the research question was very simple that how we can predict the function of an unknown protein just from its sequence. This is where the BioPython comes under. Uh, this is where the BioPython comes under this. Uh, the methodology of this, the pipeline, or we can say that. To solve this problem, I have followed a standard pipeline method of bioinformatics. Firstly, I have selected a protein sequence. After that, selecting from the protein sequence from the database, I have checked the quality of the sequence, whether it is suitable from a project or not. After that, I have performed a homology search using BLAST. Based on the BLAST results, I did functional annotations. Uh, uh, I have done functional annotations, and after that, I have came into my final biological interpretations. One of the most important thing to mention here is that the blast, which was performed programmatically using BioPython, not through the browser as required for the as required in the code project curriculum. And here, as you all can see, the sequence analysis and the homology search based on the blast results. Let me show you all the task performed in my VS Code editor. Here, my project folder is open. In the analysis sequence, I have named a file sequence Python file, where uh, this is one of the important script used for the sequence quality checking. I have loaded the protein sequence from a FASTA file using BioPython in my data folder inside my project folder. Then I calculate the sequence length to see whether it is suitable for the further analysis, especially for the blast. If the sequence of my protein sequence used is too small, uh, too short, if I say. the blast results were usually unreliable. Based on the length, the scripts write a summary into a text file, which will be documented into the quality checking summary text file in the results folder of my project run. Now, if I talk about the blast into my BioPython using BioPython, in this script, Python script, I have performed blast P, which was basically for the protein analysis, the blast one. Programmatically using BioPython here, the protein sequence is submitted to the NCBI blast server using BioPython QBlast function. Uh, okay. Instead of running this blast on the website, this code sends the request uh, to directly to the browser through Python. The results are saved in XML format, which will be directly made into this file after my blast file will be run. This step basically satisfies the requirement of using BioPython for Blast, not like browser based, but we can run it through our local based systems also. Now, if I show you the Blast results here, after the result is completed, I summarize the key Blast findings in these files. The top blast hit showed similarly to the uh, urido, uridoglycine amino hydrolase, which have a sequence length of 261 amino acids. High similarity usually indicates that the unknown protein may have a similar function. Uh, okay. Now, if I talk about the functional annotation file, in this file, sorry, based on the blast results, I have performed functional annotations. The protein is likely enzymatic in nature and seems to be involved in amino acid metabolism. Uh, it's important to mention you all that it's a computational prediction. Uh, also it is known as like in silico analysis. In other terms, experimental validations should be needed to confirm the function. So let me switch back to my PPT first. Yeah. Here you can see the results of this is the results of my sequence quality checking, where the sequence ID is being checked and the sequence length of the particular FASTA file has been checked. And this is the output of the XML file, which has been resulted after the blast file, which was in my analysis folder, has been run. Okay. Uh, as shown earlier, the functional annotation and the biological interpretation, I have explained it in my project folder already. Okay, this to conclude this project demonstration, it shows us how Python can used to analyze protein sequences and predict their functions using a systematic pipeline. The key learning of this project is basically that the homology based analysis, which I have done computationally in my Python file using my Python file, is very powerful in the field of bioinformatics. Even when the experimental data is not available, computational tools can provide strong biological insights. That's all from my side. Thank you everyone for listening. I will be happy to answer few of the questions if raised.
Okay, Aditya, you did a very great job. I like the way you explain your project in a very simple manner. But can you show me how did you retrieve the sequence of your interest?
Uh, okay, ma'am. Okay. The data from the database itself, ma'am.
From where you retrieved your sequence?
Okay, okay. I have Okay, let me show you. One of the websites like Uniprot and NCBI were mentioned in the project for a guidance. So I have just gone through the Uniprot website and here search for the hypothetical protein bacteria. Uh, well, it is already there. It was in my cache files. So from here only, I have go to the reviewed and unreviewed one, and after that, I have selected E. coli bacteria, and after that, by seeing the length of amino acids, I have figured out that what particular type of amino acid will be fit in my project. So I have just take a mediocre one of 261, I guess. So yeah, from here only I have downloaded the FASTA file, and after that I have performed the rest of the project.
Okay, very well done, Aditya. I'm really proud of you that you completed the project and also the presentation. Not
Thank you,
everyone
ma'am.
did the presentation part, so I'm really proud of you.
Thank you, ma'am.
Okay, is there anyone else who want to do the presentation? Arundhati, Muhammad Ali, do you want to do the presentation?
Yes, ma'am.
Okay, Arundhati, you can start.
Okay, ma'am. Ma'am, if my screen is visible.
Yes, it's visible.
Good evening, ma'am. Myself, Arundathi, completed MSc in Molecular Biology and Biotechnology. Today, I am going to present a Biopython project based on homology-based annotation by using Biopython programming. In that, I have chosen 5 different hypothetical proteins from 5 different model organisms. So throughout this project, I used the loop program for running blast for simultaneously for 5 FASTA files. First step, biological sequence selection. I have selected 5 hypothetical proteins from 5 different model organisms: human, rat, roundworm, rice, and tail crates. The FASTA files were retrieved from Uniprot database by using. Sorry, ma'am, 1 minute. Yes, ma'am, I'm continuing my presentation. I am audible.
Yes, you are audible.
Yes. Next, the 5 FASTA files are retrieved from Uniprot database. The second step is sequence quality parameters. In that, I have used codes for finding the file name, sequence ID, description, sequence length, and amino acid composition. Under the quality parameters, I have identified the valid amino acids and calculated the valid amino acid percentage and their how much composition are present in that each protein with valid amino acid presence. Next, sequence filtering and validation. Under this, I have first converted the sequence into the uppercase, the uniform comparison for. Second, I calculate the valid amino acid percentage by using this formula, number of valid amino acid divided by total sequence length into 100. So I set the criteria like the minimum sequence length will be 100 and the minimum valid percentage of valid proteins, valid amino acids or 95. So by using the if, else if and else words, I have made the decision to reject the proteins which are not completed the criteria which I have given. So the five proteins which I have selected are all passed the this decision making and accepted for further analysis, homology based functional annotation. The fourth step is blast. First, I have inputted the modules NCBI WW for blast and time delay for delay control for because I am running the five blasts simultaneously one after the another. Then once the first file is completed the blast, it will give the message. After that, other files will be running the blast, and the protein sequence will be submitted to the query in the NCBI blast server, and from that the blast results retrieved. After completing the XML file will be downloaded in the Python VS Code. So I introduced a 10 seconds time interval for running each file blast. Next, from the blast result, by using blast XML result files, I imported NCBI XML code, and after that the blast results will be open, and through loop code, each blast file will be read. Selected to display these criteria: best it title, alignment length, bit score, e value, query sequence, match line, and subject sequence. Next, functional annotation. Based on the its that I have obtained from the blast results, only human hypothetical protein, roundworm, and rice tail crest, I have identified the function. But in rat, there is no it that have been aligned with a proper protein. It aligned with the uncharacterized protein only. In human, the hypothetical protein is matched with DNA JC13 protein, which is highly conserved across the greatly apes like orangutan, gorilla, etc. And in a roundworm, the hypothetical protein is similar to that of F-box proteins, which suggests that it has a big role in protein degradation. Next, in rice, the hypothetical protein matched with the UDP glucuronate xylan X glucosyl transferase, which have the functional role in xylan biosynthesis and plant cell wall synthesis. And in tail crest, the hypothetical protein was similar to that of NPR4 protein, which plays an important role in defense mechanism by involving in the salicylic acid signal pathway. The key learnings from this project: actually, I didn't know the base of this BioPython, but by understanding the classes, I understand to import the sequence and retrieve the sequence from the database, and I have run the blast, and I can able to interrupt the results, and by using the blast results, I can able to find the function of the hypothetical protein which I have been retrieved from the UniProt. So I have used these codes, and I learned to use their codes in the BioPython. From this, I have understand this much only. Further, I can able to improve my skills. Thank you.
Very well done, Arundathi. You have done a really great work. I really liked how confidently you handled multiple sequences. That was a really nice job. Very well done, Arundathi.
Thank you, ma'am.
Mohammed Ali, do you want to do the presentation?
Yes, ma'am. Am I
Okay.
audible?
Yes, you are audible.
Okay. Also, audio. Is my screen visible?
Yes, it's visible.
Okay, let's start. Okay, myself Muhammad Ali, and I'm a basically biotechnology graduate. So my project is functional sequence characterization of an unknown protein using sequence analysis and homology based annotation. So this is my presentation overview. This goes like this: first is project overview, then the six step pipeline, then sequence analysis, quality, sequence quality analysis, and then homology search results, functional annotation, biological interpretation, conclusion, and key findings. So moving on to our project overview, it has basically three things: core research, questions, project goal, and pipeline approach. The research question is: given an unknown or hypothetical biological sequence, how can we computationally analyze to predict its quality, similarity, and biological function? Which basically means that if unknown sequence is given to you, what basically tools can you use to do to check its quality, similarity, and biological relation with the species, different species available in our data sets. So for the our goal is implement a complete bioinformatics pipeline in computational analyze computationally analyze an unknown biological sequence, predicting its quality, similarity, and biological function through systematic sequence analysis and homology based annotation, which is basically our six step pipeline approach, which we discuss further. This is our six step pipeline workflow. For the first step is sequence retrieval, the second step is quality analysis, the third step is filtering and blast, filtering and validation. The fourth step is blast homology search, and the fifth and sixth step is functional annotation and biological interpretation. This workflow is important because each step feeds into the next, creating a comprehensive analysis pipeline, which means that you cannot directly jump to the last step without completing the previous steps. So our first step was sequence retrieval, for which I used Uniprot for obtaining my hypothetical protein sequence from E. coli, which is my chosen microorganism. So the moving on to its second step was quality control analysis, for which I checked its sequence quality, which is GC content, ambiguous residues, or open reading frame completeness, and the length of the sequence, which is 505 amino acids. So our decision was to that it is suitable for downstream analysis. So our third and fourth step is filtering and homology search. The third step is filtering and validation. For that step, I decided that my sequence must have 300 amino acids, no ambiguous residues, complete open reading frame verifications. So the fourth step was running blast homology search. For that, I used Biopython to run send my sequence to NCBI to run protein blast. The results are that my sequence matched to DGTPase, in which is found which is an enzyme found in E. coli, and it matched 100%. The E value is 0, which is highly homologous. The DGTPase family, it belongs to DGTPase families. So the moving on to our fifth step, which is annotation summary, which is important that it tells why the what the sequence is related to, why it is needed, and and what functions it play, why is it important. So moving on to its first, that it is ggtpa triphosphohydrolase activity, which is basically breaking down the water molecules for DGTP process, which is nucleotide metabolism pathway, using nucleotide metabolism pathway. And EC number is enzyme classification number, which is 3.6.1.15. And our confidence is that it is highly identified. So moving on to geo terms, which is gene ontology terms. Molecular function are DGTPase activity, hydrolase activity, and metal ion. The basically nickel ion it uses. So the DGTPase activity is the incorporation and breaking down of DGTPase in the DNA of E. coli. Hydrolase activity basically is breaking down of H positive and OH molecules from water. Biological process is NTP catabolic process, DNA metabolism, DNA replication, and it is found in cytoplasm and cytosol. Our blast results shows that the five hits are the top five hits are the highly matched. The first one is the exact match, which is 100%. So the remaining are complete matches, but there is some excessive amino acid with them. So for that purpose, it showed 85%, 78%, 72%, 62%. Query coverage is 100%, subject coverage is also 100%, gap is zero, mismatch is zero. The perfect alignments provide definitive and evidence that our sequence is identical to characterize DGTPase from E. coli. Identified protein is an enzyme, which is DGTPase enzyme, where it has full name of deoxyguanosine triphosphate triphosphohydrolase. Why we think so? Because of its 100% identity, sequence identity from the E. coli K12. K12 is a strain of E. coli. E value is 0, indicating high extremely significant homology cross conserved cross enterobacteriaceae family. Multiple PDB structures available, which I'll show you at the end. What evidence supports this claim, which is again the PDB crystal structure, protein structures? Functional studies demonstrate that it is highly substrate specific when we use it in our labs. So, and the genetic evidence shows that if we somehow knock out this gene from E. coli, it causes its death. Evolutionary conservation shows that it is very important for the survival of E. coli, and it is highly conserved across whole Enterobacteriaceae family. Moving on to biological significance and evidence, which is again biological functions. DNA repair prevents mutagenic DGTP incorporation during DNA replication, maintaining genome integrity, which is that it incorporates DGTP, which is basically a nucleotide, into the DNA of the E. coli. Nucleotide pool sanitation. The second function it provides is the proper balance by hydrolyzing excess GGPT, preventing toxic levels. Meaning that if the environment has excess GGTP, it will break them into its constituents. Antimicrobial target, potential target for antibiotic development due to its essential role in bacterial survival. This is again the same that it has crystal structure and active site, in vitro Functional study shows in vitro enzyme acid demonstrate its function as a hydrolyase enzyme. Genetic evidence shows that highly conserved deletion results in lethal evolutionary conservation, highly conserved in whole E. coli family. So moving on to our project requirements and tools. The first requirement is Biopython. The second requirement is NCBI and BLAST. The third requirement is Uniprot, and the most important is having Biopython library. This is our overview of the project. These are the files. So moving on to the conclusion, the analyzed sequence is definitely identified as DGTPase from E. coli with 100% confidence, playing a crucial role in nucleotide metabolism and DNA replication. Our key takeaway is that this project demonstrates the power of computational bioinformatics pipeline for rapid and accurate sequence characterization, transforming unknown sequence into biological meaningful insights using freely available tools and databases. For this project, it means that for this project you don't have to use any paid uh, tools and databases to complete this project, and the pipeline is also important because of its workflow that the next step feeds the previous step feeds into the next one. Moving on to my, I'm showing you the structure of this is the crystal structure of ggpts ggtps. Here it shows with two DNA effector molecules. Let me show you. You see this gray line, it is the DGTPase and DNA collaboration, which is this is DGTP1 thiol, which I showed you, which I told you about in my presentation. And this is for the nickel. You see this is what nickel is needed for. So thank you everyone for listening.
Ali, your presentation was very nice.
Thank you, ma'am.
Also, the flow of your explanation was very good. So, can you explain your code as well?
Ma'am, this is my input sequence. So this is my code which I have shared as in my form. These files are available on GitHub. So these are the first three steps. So for this I use the XML file, and this is the long code for writing annotations.
Yes, Aditya, do you want
And
to ask questions?
this is the
Yes,
XML file, and this is the README file.
Okay, Ali, Aditya wants to ask you some question.
The house is yours, master.
Yeah, good evening.
Good evening.
Uh, I have just one doubt. Like, while you are presenting your presentation, you said that at ending, using this particular project, this type of tool, we will not be having a need of any paid tool or database. Okay. So, first of all, like it was never about any paid tool or anything, as the blast fee is already free for us on NCBI. So, how you would you like to like tackle your own works?
Basically, when you go to different institutions, they tell you that they have some purchases, they have something, they have to make some purchases from the database. Says it was to tackle this question.
Okay, well, it hasn't satisfied me yet, but okay, I will let it go. Like it's a very generic question. Sorry, but yeah, I have to ask it because somehow you were like missing the flow of connecting the dots. That's why I had to ask this.
Okay, okay.
It's okay, Ali. Your presentation was very nice. Also, Ali, I saw that you performed the GC content.
Yes, ma'am.
Can you explain me why the analysis of GC content is important?
Ma'am, basically to check whether it is our structure is stable or not stable. When it has passed a criteria like 30 to 70% of GC content, in GC content it shows that it is a stable molecule, so we can move further with that.
Okay, very good, Ali. Okay, so Aditya Panigrahi, do you want to also do the presentation? Excuse me, Aditya, I didn't understand. Do you want to do it?
Yes, ma'am.
Okay. Ali, very well done. Okay.
Thank you, ma'am.
Okay, Aditya, you can start.
Is my screen visible?
Yes, it's visible.
Okay, myself Aditya Panigrahi. I am from BTech Biotechnology, SRM, IST Ramapuram. My project is based on a bacteria. It's like the outer membrane of a bacterial protein. I'm going to discuss about this because I have been working on this bacteria like during my phytochemical research as I'm working on it, so that I choose this one as the easiest way to go with tackle this project work because I am going to use those things itself in my research paper also. Uh, so let's start it. Yeah, my chosen protein sequence is P68208, which is Shigella flexionari, which is a gram-negative bacteria. It's an anaerobic, so a non-motile bacterium. So we went through some of the things like microscopic visualization. This was done in my college itself while I was going through. It is image J, right? Everyone must be knowing because everyone is from biotech department itself. So in image J, if we put some of the images of some bacteria, so we can go with uh, the visualization under the microscope. So after taking the picture, we can put it on that website or that software so that we can get the thermal or any kind of images like what are the activities going on. So I used this that image for finding this out. This is from Pinterest itself. So this is what that is happening. How the feces of one person, like going outside and doing toilet and all outside. So some of the people don't wash their hands, then their fingers, then flies go on that, or people working on fields, they might carry out those bacteria with them. If they don't wash their hands, that it will go to the food and they will consume some food, then they will be causing that bacterial infection, so that they will be either having abdominal pain, fever, or diarrhea uh, with blood and mucus itself. So treatment is antibiotic only through oral intake therapy, but there is no vaccine on it. So the person has to recover by itself only. So new host can be taken, means one host is doing the thing, then the next host, if that another person is coming to that same place, then they are doing the same thing over there, then it will be transported to the other person itself. So global burden is like 2 lakh deaths for every year. So it's very high. So on that, like phytochemicals, I am working on neem, moringa, and bay leaves. So it is a both kind of like wet lab and dry lab working. I have not gone through molecular and all because it's Biopython only, so no molecular docking procedure. So in this, this is from Uniprot itself. I have gone through like while going through it. So it is a amino acid of 69 amino acids, which has about 7.7 kilodalton molecular weight. Its biological relevance and all. Then the sequences and its retrieval. Okay, let's go to my VS Code. This is my first step. Okay. In my first step, I am doing my sequence retrieval. At this step, I went to my Uniprot website. I searched for the name. Then after searching for the name, I got through like P68208 is having a shorter amino acid sequences, and on which I can work out in a very short span of time because it was a very rush time itself for my college itself. So I went through this. So this is my code for the first step, which I had performed for biological sequencing, in which I got that the FASTA files properties, like its ID, record ID, number of amino acids, and the protein sequence that I had found. I will show you the FASTA file that I had downloaded. This is the FASTA file. This is the protein sequence that I am working on. Then the second step, in this step, I went through like sequencing quality and analysis. In this step, I went through some of the things like alkaline counts, cytosine counts, glutamate counts, and all. So these are some of the sequences like what to say. Some of the chemical reaction, like what are the chemical properties present in my sequence. So I wanted to find it out. So I will find the result also. Yeah, so in this, I found that there is no stop codon in the sequence, and my expected was also zero only. So I went through those datas. like how much is the number of counts in the sequence, that is tyrosine and all, valine, whatever we learned in our 12th grade and first year itself. So I used those sequences only. So I didn't go much deeper because I didn't know much more about this because I'm second year only. So the next thing is our quality analysis, in which I did my filtration and all. So I filtered my FASTA file first. So I will run this out. Okay, my run file is this. Where is the filtered one? Yeah, this is my filtered one. Filtered YGB, as it is shown. It is saved in this form. So this is the sequence that is after filtering my QC part. It is QC passed already. Then the next thing is fourth step, the homology analysis. In this, as during my lectures, like how ma'am taught me to go with the like how to find the blast files and all. So I ran this sequence. This may take like about 2 to 3 minutes. So I have the result with me. That is blast result. In this, there is about 1 minute. Hit number is 50 over here, and our HSP is also found for every sequences. So as we know, we went through all those things. Like the blast file is present here. There are about 1,441 uh, HSPs and hits which are present. After the hit, there is an HSP, as we know, that is high scoring pairs. Then the next step is parsing blast results. In this file, I went through all those things like the steps of parse, blast XML, then loop files by aligning all of these key datas, then HSV high scoring segments pair, that is by finding all those things. Uh, I will run it out. Yeah, so in this file, I got my e values, scores, that is about 175.252, then their query reason and the subjects and all. There are about homologs of 44 in this area, that is XP underscore 053418912.1. It is about insulin and all. So I will show you another thing that I found. Yeah, in this we found that our e values, as man told, that e values are very important. These are the e values, and the query recovery are like 100. Those which are taken as 100, I used those 100 only during my coding sequences and all. So these are the homologs. Then homolog retrieval. In this, I went through if there is any error in my code. So I ran this. So I don't get my result as wrongly proved. So the identity is about 99.09%. This gene bank shows. So e value is also given. So with this retrieval, all have been fetched out. The sequence is successfully saved also as homolog plus. This is a sequence that I have found in my result itself. So these sequences are similar to the sequence that I had used uh, prior for my protein sequence as my project and all. So this is Homo sapiens like in humans, in gorilla, and synthetic construct and all. So these are the values and all. So then the next is the result that I had found out from the whole thing is the sequence retrieval. First, that is 69 amino acids are there, about 7.7 kilodalton long. Then it's 48% percent about of sequence quality analysis, that is high hydrophobic content. So we went through some of the like faster files generation, then blast analysis, in which I found that e value which is less than one e or t, which has about 95% of its identity that has been used for the then the functional domain and all like trypsin, the values of AA 66 and 242. This 66 242 shows that the in the blast file the number of the area in from which the code is taken. So between 66 to 242 it is taken. So the 2 is PDZ, which is about AA 269 to 337, and 364 to 454. This is the analysis. Then the conclusion is this. So we are working. I am working on this. So the stress resistant in this function of protein quality control. I would like to thank Muskan ma'am for teaching us Biopython and making me knowledgeable regarding this Biopython thing, because I didn't know that Python can be used in biotech field also. Completely like I used to use in molecular docking and all. So I didn't know that Python has a very vast thing in it. So I'm very happy that I went through this course. Thank you, ma'am.
Okay, very good, Aditya. I want to ask you one question.
Yes, ma'am.
So you said that your sequences does not have any stop codons, right?
Yeah,
So can you tell me what do you mean by that?
ma'am. Stop codons is like while making DNA sequence, right? Like in plasmid and all, while making the DNA sequence, we are having our leading strand and lagging strand in it. So leading strand contains what to say, SDS content, which is not given for preventing annealing and all. So during that procedure, there are some of the like pyrophosphate, right? PPI, so pyrophosphate and all. So when pyrophosphate comes and binds with another sequence of nucleotide, when it synthesizes itself to make its DNA, so there is a stop codon, like there is a protein sequence which makes the lag, and telomerase reaction occurs, due to which there is a high workout of this cell, like workout of nucleotides. The thing is, when telomerase reaction occurs, like telomerase reaction occurs, during that time the daughter cell is shorter than the parent nucleotide. So when there is a telomerase reaction, then another telomerase one sequence will be attached to it. So due to this codon only, there is no action of the what is it, the nucleotide actions. Like the nucleotide will not grow along, like it will not grow out
Hmm.
because of that.
Okay, so if a sequence does not have a stop codon, will it? will it be still be considered a full length gene?
No, there
Why?
should be a stop codon.
Yes, okay, very good, Aditya. Your presentation was very good. All of you did a really good job, especially Arundhati with the
Thank you,
multiple
ma'am.
sequences, and Aditya, and both of your names are Aditya. You did a very good job. Ali, you did a very good as well. Okay, so you have filled out the form for the certificate.
Yes, ma'am.
Okay, all
Yes,
of
ma'am.
you?
Yes, ma'am.
Okay. Okay, everyone, have a good evening.
Good evening, ma'am.
Good evening, ma'am.
Good evening.
Good evening, ma'am.